I'm Jack Kerrigan. I'm the director of the pediatric epilepsy program at the Barrow Neurological Institute at Phoenix Children's Hospital. I've had an interest in treating the hypothalamic hamartoma patients for the better part of 15 years, and I've had the privilege of dealing with many patients with it. And I'm a founding member of the Medical Advisory Board for Hope for Hypothalamic Hamartoma.
I'm Heinzen. I'm a geneticist at Columbia University. I do research into epilepsy genetics primarily.
My name's Sam Berkovic. I'm head of the Epilepsy Research Centre at University of Melbourne in Melbourne, Australia. I'm a clinical neurologist, but I work on the genetics of epilepsy with collaborators like Erin. I've had an interest in hypothalamic hamartoma since my fellowship in the mid-1980s in the Montreal Neurological Institute, and have kept seeing patients and thinking about the biology of this disorder since then.
Okay, so basically the clinical issue that we were after is what's the cause, what's the originating cause for the HH problem. And the clinical clue that we had is that patients with Pallister-Hall syndrome who have other problems, but they're known to have a mutation in the GLE3 gene. So that basically set up the question, could the patients who have the sporadic form, meaning most of the patients, 95% of the patients with HH, could they have a mutation in the GLE3 gene, but a somatic mutation, so only in the tumor? So a lot of the genetic testing that we've done over the last several years, and specifically this most recent study, addresses exactly that issue. So basically it's comparing, it's searching for a mutation that's present in the tumor, in the HH lesion, that's not present in the blood. And that can be the clue that would tell us whether that could be a gene that's responsible for the problem.
We performed genomic analysis on the hamartoma and compared the genomes of the hamartoma to the genomes from the blood cells, and to identify mutations that were specific to the hamartoma.
So I think it's been incredibly valuable to bring in the multidisciplinary group of experts. I've learned a lot about the development of this lesion just from this conference today. The area of the brain that it's in is incredibly complicated in terms of its development and its embryology, and having experts to come and discuss this in the context of understanding the lesion has been extremely exciting. And I'm sure we'll all go away from this conference and scratch our heads a lot harder and think a lot more deeply to get a better understanding of what's going on, and potentially thinking about new ways to approach it.
You know, it's hope for the future, because without this kind of understanding, we're really at a lack of getting at the basic mechanisms. You know, why does this particular tumor cause seizures? You know, how does it have an impact on the rest of the brain? And so this kind of genetic discovery doesn't immediately lead to a new treatment. But if we're going to get to that kind of curative treatment or that breakthrough moment, it's going to be with this kind of research.
Well, I think in all research it often kind of lights a flame of taking things further. So we need to do further and larger studies to understand the genetic side at even a deeper level. We do understand it a lot better in 2016 than we did five years ago. But the next step, or the translational step to understanding how we might turn this into better treatments for patients, is going to depend on much more studies in the laboratory, ideally animal models, which are challenging to develop. But we do hope that that might be possible and that new therapies might emerge.